Welcome to everyone. Just would like to get started with the webinar today. So on behalf of the JPIAMR Initiative on Antimicrobial Resistance, I would like to welcome you warmly to the launch of the JPIAMR Virtual Research Institute Digital Platform. So I'm really looking forward to today. My name is Laura Marin and I'm the head of the JPIAMR Secretariat. So today we've been looking forward these days for a long time. It has been a few years back that at the board meeting of JPIAMR with all our members, data sharing and data accessibility to the AMR research community. And after that, it came the idea to develop a virtual research institute. And from there on, we've been engaging with all the member states, with a lot of people, setting up a team who has been working very, very hard in developing what we are going to present to all of you today. So welcome to the webinar. We are going to get started with some presentations about the concept of the JPIAMR Virtual Research Institute, and as well as a demo of how the platform looks like and the usability and how to use it and what is the value for you. And then we are going to move into a very exciting panel that is going to be focused the discussion about the value of interlinkage of data and integration of data sets. So that's what is on stock for the next hour. Just to let you know for today that this webinar is going to be recorded, and then we're going to share with all of you and with the wider community in our JPIAMR YouTube channel. And throughout the seminar, please use the Q&A function in Zoom, where you can post questions or input or comments throughout the webinar today, and we will deal with your questions after the panel session. So please feel free to use it throughout all the time. For all of you that are not that familiar with JPIAMR, so we are a global organization that at the moment it comprises 28 member states across all the continents, and also the European Commission is a member. And what we believe is that science has a central role in tackling the societal challenge of antimicrobial resistance worldwide. So our function, our main mission is really to support the scientific community, to support science, to bring new knowledge on the forward into solutions directly for the citizens and the patients regarding antimicrobial resistance. What we do is basically we coordinate research funding at national level and international level on antimicrobial resistance, and we jointly fund and support science, and as well as we prioritize science and make an interface with the policy level on implementing those solutions. So it's all about global coordination and interaction. How we do that? So basically, we have developed jointly together at the global level a strategic research and innovation agenda on antimicrobial resistance, which has a One Health approach. And that is the research areas, the priority areas that we are supporting in this initiative. So it goes from therapeutics, diagnostics, surveillance, transmission, environment, and intervention. So it's a full scope on antimicrobial resistance that we support. Up to now, in the last years, we have got a total investment of over 100 million euros through competitive calls. And we have at the moment a portfolio of over 100 projects and across 71 countries, and supporting almost 1,300 researchers worldwide as PIs. And this includes a much further scope than our membership. We also have funding mechanisms to support lower middle income countries. So this is all how we support science in a way of competitive calls, but beyond that, we will also like to support to bring forward AMR knowledge and new treatments and new interventions through tools and capacity, increase the capacity in this community. And if you check our website, so we have an AMR knowledge hub where you will find tools and resources in it, from research publications relevant to the different areas to a full dashboard with all the projects that we are supporting worldwide, with all the details on those projects, and you can get also on the level of investments in each area. And also we have like a map of research infrastructures on AMR and list of biobanks, etc. So I welcome you to check all those. So the next step in this effort that JPIAMR is doing on how to support the community has been to really move into integration of data sets and accessibility and sharing of data. So therefore, we have developed what we're going to present today, this digital platform, which is really a unique entry point of interlinkages of different data sets. And from here, I'm going to give the torch after this introduction to Till Bachman to move forward on the webinar. Till is has been chair of molecular diagnostics and infection and the deputy head of infection medicine at the University of Edinburgh. And he has his PhD from the University of Stuttgart and the University of Tokyo, and is an expert in point of care detection of infectious disease and antimicrobial resistance. And most importantly, for me, most especially, is the current vice chair of our scientific advisory board of JPIAMR. So thank you for everything, and also for being very committed to this process. And please, the floor is yours.
Thank you very much, Laura. And as Laura said, I've been involved with the VRI and was very happy to be part of it, and really excited about the day-to-day, about the launch, and very happy to do the moderation. And we have a fantastic panel in front of us. So, so it's my honor now to introduce our first two speakers, and for the presentation, for the introduction of the VRI Digital Platform, and for a demonstration. So yeah, very happy to introduce Edith Brochu. Edith is the really driving force behind the digital platform and the VRI. First time I heard Edith speaking about the VRI, I was really intrigued by the enthusiasm and the opportunities presented through the VRI and then the digital platform. So I'm really glad we are here today and hear Edith speaking and introducing it. So Edith is the Project Manager Strategic Research Initiatives at the Institute of Infection and Immunity at the Canadian Institutes of Health Research. And by the way, you have all the biographies of the speakers on the website. So Edith has a background in experimental medicine from Laval University, and has experience in working in pharma industry as a researcher herself, and also with non-profit organizations, and a background in oncology and personalized medicine. So Edith will give us an introduction into the VRI, and will be followed by Martin Adrians and from ONTOFORCE, who gives us a live demo of the virtual platform, of the digital platform. And Martin himself, he has a bioscience and engineering and bioinformatics background from Leuven University, and he has a very long experience also working with life science industry. So together, very good team in presenting this. So Edith, floor is yours for the introduction of the digital platform.
Thank you, Tal. Can you confirm you see my slide, Tal?
Yes.
Thank you. Good morning, good afternoon, and good evening to all. It's with a profound pleasure that I'm here to give you a bit of the background information on how we came about developing the Virtual Research Institute and its digital platform for JPIAMR, or the Joint Programming Initiative on AMR. So as you can appreciate, the AMR field is very active and is also multifaceted. It's covering the entire One Health spectrum, and it's requiring the involvement of stakeholders from various disciplines around the globe. So as Laura mentioned, when we first started to discuss the development of a Virtual Research Institute many years ago at JPIAMR, it was because we wanted to make a mark in strengthening all the international collaboration in AMR. So it's with that in mind that JPIAMR was happy and agreeing with the statement from the 2019 Interagency Coordination Group report to the UN that recognizes that AMR research is in dire need of global alignment, coordination, and better infrastructure to strengthen collaborations, facilitate innovation, avoid duplication, and increase knowledge exchange. So therefore, the reasoning behind creating JPIAMR-VRI and its digital platform was to that AMR research was creating a plethora of very extremely valuable data. However, most of it was unfortunately siloed, inaccessible, or invisible to the global AMR community. So we thought what we need is actually a platform to house all of that efforts from the AMR community and under a structured and coordinated roof for the benefit of all. JPIAMR-VRI wished to put down barriers in the fight against AMR. So we want to do this by promoting the One Health response and connecting the right people to the right data. Also by maximizing access to the research result worldwide and speed up innovation. By reducing duplication of work and generating catalytic movements and more synergetic investments. Also by creating cross-pollination between all sectors, disciplines, and ideas. And by accelerating the development of new resources, connecting all stakeholders involved in developing new solutions. We operate under 4 mandates. The first one is to connect people to data. So that is to facilitate data and information mining and connect users to the resources. The second mandate of the VRI is to connect people to people with more interactions, more relationships, more networking, and more visibility. Mandate number 3 for the VRI is to connect data to data. So linking siloed data and data sources to support real-time insight and knowledge exchange at the global scale. And lastly, the 4th mandate of the VRI is to encourage better mobilization around the AMR fight and help keep AMR on the global agenda. We built the VRI vision around 8 distinct components, or the rooms of the house, if you prefer. Those will be or are the key elements to achieve the 4 mandates I just mentioned. So if we start here around 8 o'clock here, we have the partnership and collaboration room, or the component that facilitates partnerships and help build those academic industry relationships. Then we move to the research project hub in blue. That room is to know more about the funded projects in AMR at the national and the international level, while also learning about the outcomes and outputs of those projects. In pink up there, we walk in the research funding data hub. And this is to bring some critical information and funding opportunities around the globe, as well as what the research investment landscape looks like and where are the gaps. In navy blue, we open the research mapping room. That is to look for information on who's doing what, where is that, where are the various technological platforms, and what are the specific expertise that I'm so much looking for to continue my project. Then the capacity building, that's about learning opportunities, conferences, training and teaching, summer school, and all of that capacity building material. Then communication. The communication component is, like I like to say, the kitchen counter island. That's where we make that people-to-people connection. And it's also where we increase awareness and we disseminate knowledge. Then in light green here, we have the resource such as the guidelines, the best practices, reports, and policies. And last but not least, we have the data knowledge sharing hub. And that one is to access the valuable AMR-related database, catalogs, and libraries, as well as tools. So that house could not be built up in the air. It required a very solid foundation. And that foundation came with the help of the JPIAMR-VRI funded networks. And we are honored to have the coordinators of both the AMR-DX Global Network with Dr. Bachman, as well as IRAD Network with Dr. Mueller here today with us. We also had various consultations with AMR stakeholders, including a hackathon, and learned a lot from the response received through the needs assessment survey. That survey was ran in spring of 2020, and it showed that amongst the 8 components of the mind maps, the data knowledge sharing hub was cited as the top 1 development priority 3 times more than any other rooms in the house. So that's where we knew we had to look for an experience builder for our digital platform. So we associated with ONTOFORCE and their existing Discover platform. We are therefore launching today with ONTOFORCE the JPIAMR-VRI Digital Platform, a free access point to a collection of data sets for the entire AMR community around the globe to facilitate knowledge and data sharing and to support continuous dialogue and multinational collaborative research amongst AMR stakeholders. The digital platform integrates, with approval from the data owner, carefully selected relevant AMR-related data sources with more than 145 different public data sources already available on the Discover platform. So this will help the user analyze, integrate, and extract AMR data more efficiently and bring simplicity to complex data sets. I will only briefly present to you those 5 data sets now, because I know that my colleague Martin from ONTOFORCE will cover them all in details, but also that we have with us today as panelists, Dr. MacArthur from the CARD and datasets, and also Drs. Colino and Madec of RISAPAT, which I'm sure will have a lot to say about their own respective datasets. So let's start with the first one. We have CARD, the Comprehensive Antibiotic Resistance Database. That's a database on resistance genes, their products, and associated phenotype. We have RISAPAT for monitoring of AMR pathogenic bacteria isolated from animals in France. We also have ATLAS, the Antimicrobial Testing Leadership and Surveillance Database, and that's a database that's created by Pfizer. It's about and on emerging bacteria and fungal resistance patterns and sensitivity to antibiotics. We have the NCBI Isolates browser, which identifies bacterial genomes with AMR genes. And lastly, we have JPIAMR supported projects and networks, which is allowing to have information on investments on AMR in the AMR research priority areas and search through funding organizations and countries. So now that these 5 data sets and the 145 other ones in Discover are available for free to the broader audience, one can explore the multiple integrated data source in the digital platform to find possible targets of antibiotics molecule, or to look for chemical information on antibiotics and its related biological assay data. You can use the digital platform also to look for determinants of antibiotic resistance or find resistance mechanisms of organisms or resistant genes and their sequence. The digital platform can be used to look for clinical studies, patents, publications. You can use it to look for key opinion leaders in the field of AMR research, research organization and academic institution involved in AMR research all around the world. So what we are presenting to you today is what we identified as the proof of concept phase 0 of the JPIAMR-VRI digital platform. And it was made possible thanks to the support of the JPIAMR member states, as well as all the researchers and other stakeholders that were consulted. And now, before I leave you with Force and Martin for a live demonstration, followed by a panel discussion with our distinguished panelists and moderator, I'll conclude by saying that beyond this phase 0, JPIAMR is also planning on implementing more key external related data sets, AMR-related data sets, and of course, building on the feedbacks received by the AMR community about this proof of concept. So we invite you in. You have a look at the digital platform. Come visit your house. Visit all the floors and all the rooms. Tell us what you think. Tell us what you would like to see next in your house. It's yours. Thank you.
Thank you very much, Edith, for excellent presentation and opening of this session. And I very much like your ending with the, it's your house. everyone who is listening and everyone who is listening in the future, it's your house, because you have a possibility to contribute and use what's in there. So as I said, I think it's an excellent opportunity for us to exchange data and communicate. Martin.
Yes.
I would hand over to you if you're ready.
Yes, that's perfect. Thank you very much. Thank you for the introduction, Till, and thank you as well for the handover, Edith. We're very excited to start showcasing the actual digital platform. So I will start with sharing my screen. And if someone would be able to confirm that you can see my presentation.
Yes, we can see your screen.
All right, good. So what I want to do today is, of course, dive a bit deeper into the JPIAMR-VRI digital platform that was created as a collaboration between JPIAMR and ONTOFORCE. I will follow with a short introduction on the platform, what exactly is behind the platform as technology, what is the platform, what are the capabilities, and then also followed by a live demonstration of a couple of exemplary use cases on the platform itself. Of course, as Edith already mentioned, the aim is to improve efficiency and coordination of antimicrobial research across all these networks and facilitate knowledge sharing and knowledge exchange. With that being said, the platform on which JPIAMR-VRI Digital Platform is being built is Discover, and Discover is developed by ONTOFORCE. Discover itself is a self-service knowledge discovery platform that enables smarter data-driven decisions and accelerates time to value by unlocking insights from all these siloed data sources, as Edith already mentioned. What this means is that data from very different places can be brought together in one centralized platform. With additional power of semantic web technology and also linked data technologies, topped with an intuitive UI, we want to bring any type of user the capabilities of retrieving information and value from all of these data sources. As Edith already mentioned, the 5 data sources were selected according to the correct data sharing principles in collaborations with the respective parties, for which we are very grateful. As already mentioned, we have CARD, Comprehensive Antibiotic Resistance Database. We also have the NCBI Pathogen Detection Database. It captures a lot of isolate information with specific to antimicrobial resistance, stress responses, virulence genes, and a lot more information. Then we also have Atlas, which is sponsored by Pfizer, the Antimicrobial Testing Leadership and Surveillance Database, and it's openly hosted by the Wellcome Trust. And this, of course, also offers a lot of extensive data on bacterial and fungal resistance patterns. Then next to that, we also have ResoPath. ResoPath is the antimicrobial resistance surveillance network for pathogenic data isolated from all kinds of specific animal species. It is, as mentioned, coordinated by ANSES and brings data from all across laboratories across France. And then lastly, the JPIAMR supported projects and networks was also integrated here, and which allows the user to browse and extract knowledge from them and even maybe use these data sources to find possible partners for future projects. I've also added a ... data source, of course, to highlight that in the future, other data sources can also be added as well. As mentioned by Edith, ONTOFORCE, we as ONTOFORCE provides additional public data sources, more than 145 at this moment, capturing a whole wide range of data across the full medical sector. The goal of the project was then to combine this already existing model that we have on this public data, combine it together with more AMR-related models, and enrich this public information together with more of these AMR-related data sources but also enrich public information with AMR-related data. Good. With this short introduction, I would like to start a live demo and showcase this data integration, followed by a couple AMR-related use cases. So let me switch over to the JPIAMR Digital Platform, where I'm presented with a login screen. And of course, at a later stage, all the access requirements will be shared as well. After I log in, I'm presented with a start page. I will come back to that right away. But what I want to do is first dive a bit deeper into the data model. As can be seen here, you'll see that there's a list of all the public data sources and all of the additional data sources which are AMR-related that we have integrated into the system. As you can see here, the location called public is actually our public information. And then we also have the in-house datasets which are integrated here: CARD, RISAPOV, JPIAMR dataset, NCBI pathogen isolates, and atlas data. In the ontology graph section, you'll get an overview on how this data is actually interconnected. Based on the data sources, we can also highlight in a quick fashion how the additional data sources that are AMR related, how they contribute and extend the current existing data model, and how the existing data model actually contributes to these AMR datasets. So for example, if I select these datasets, you can see that they will be highlighted in yellow. This means that they contribute with data to this knowledge graph, and also the public information will also be extending on these AMR-related datasets. I can, for example, dive a bit deeper into one of these data types. So for example, if we're looking at active substances or drugs, we can see and receive an overview of which data sources all contribute to this specific data type. On top of that, we have a lot of information on how this data is connected to other siloed data or other public sources in the medical sector, and specifically on this platform related more towards AMR research. To come back to the start page, upon initial login, the user will be presented with the start page. They will have different buttons where they can find more information on what the project is all about, the launch of the digital platform, the different data sources, what they actually capture, with of course the necessary reference to them. And there's also the additional help page, which contains a lot of information and an extensive user guide and some video material to support the user in their journey on the platform. Good. That's a small introduction and a quick introduction to the ontology or the data model behind it, how the platform actually works behind the scenes, and how this is beneficial to the AMR research community, but also to public sources that will be enriched with this AMR data. With that being said, I would like to jump into the use cases. So I will go deeper into a couple of use cases. There's a lot more capabilities, of course, but we selected a couple of use cases specifically related to the AMR research community. And the first one that I want to highlight is how the platform, the digital platform, can help in chemical research. Let's say a researcher is interested in finding more information on a specific antibiotic. He or she would like to find more information on antibiotic targets, resistance mechanisms, AMR patterns, publications, patents, and much more. The researcher can do this by searching for the antibiotic and browsing the data model that was presented to you at the earlier stage of the demo. The retrieved data will be a combination of all of our public data, as well as the additional AMR-related datasets. which he or she can interact with using extensive analytical capabilities of the digital platform. So let's dive deeper into the first use case. Let's say a user or a researcher is interested in finding more information on specific antibiotic molecule, as mentioned. Let's take, for example, oxacillin. Of course, any other antibiotic molecule can be searched for as well, and the use case will be represented in the same way. He or she can start with a general search, which will return results across all of these different data types. For example, I can see that there's 5,000 publications coming from all of these public sources, for example, PubMed, which immediately show results and find hits for this term. For now, let's dive deeper into the antibiotic molecule information. When diving or clicking straight on these exact hits, I will be presented with a dashboard and I will find more information on my exact hits. As you can see, when toggling on the data source provenance, I can see that there's CARD, Atlas, ResaPath, NCBI data, and much more contributing to this information on this chemical or antibiotic molecule. Of course, we have the additional data sources, but we have Campbell, CABI, and much more data sources also contributing to information. This public information can also be accessed by switching, for example, to this one, where you can find a lot of more information regarding structures, regarding all kinds of information, pharmacologic actions, diseases, and so on, all related to this antibiotic molecule. When switching back, we can also follow links to all the other additional data sources that were integrated in the platform. So for example, we could dive a bit deeper into the antibiogram data provided by ResaPath. So what we can do is we can follow the link to the ResaPath data. And what this will do, this will bring you to all of the antibiograms from ResaPath, from the French community, which have a lot of information more on these antibiograms and how resistance is evolving in this case. Of course, next to ResaPath, there's also the possibility to follow and find more information on all kinds of specific isolates. And we can follow the link there, and this will bring you to a combination of Atlas data and NCBI data, which describe these isolates with more information on global resistance and intermediary data sets, with also a lot more information on the genetic aspect as well. The researcher could also be interested in finding more information which is coming from the CARD dataset. For example, he or she would be interested in finding all the determinants of antibiotic resistance, for example. When following the link, the user will be presented with the results that are related to this antibiotic molecule. Another example could be that the researcher is interested in antibiotic targets. You could follow the link, have an overview on what exactly this antibiotic target is, find more information on it, and even if it would be relevant, continue their search from there. For example, they could have a look at all the antibiotic molecules that have the same antibiotic target. These are just some exemplary use cases with additional data sources that the AMR integration brings. Of course, on top of that, we have a lot more information coming from all these public data sources. For example, we can follow links to patents, publications, clinical studies, genes, and much, much more. Good. And this is concluding my first use case related to chemical research. In the next use case, I would like to highlight the flexibility of the platform as well. where, for example, a researcher would be interested in finding more information or what is known in regards to a specific topic. The JPIAMR-VRI Digital Platform allows to apply complex search queries to find very specific results. As an example, he or she would like to find information on a hot topic, such as the impact of COVID-19 or in general a pandemic on antimicrobial resistance. Let's switch back to the platform and start a new search and create a more complex query. So for example, I'm interested in finding information on AMR, or maybe also use the filter. And the combination with COVID-19 or pandemics. And maybe let's add an asterisk behind it, just so that we can also include Burn. Straight out of the box, we receive results across my more complex search query. But the platform also allows to broaden your search, to enrich it even more with the use of synonyms. So for example, we know that there's a lot more synonyms for COVID-19. So say I'm interested in also including all of these synonyms. I simply click the button and my search will be extended with more results that are related to these to the search term. As you can see, we can find and dive straight into the results of our query. And for example, look at the 655 publications that match my results. We can, of course, at any stage, continue interactively filtering down for specific dates, start date that you would like to have a publication year for these publications. The platform also, for example, allows you to stay up to date with the results related to this topic. And this can be done using the Watch Dashboard functionality, which can be found here at the top. When clicking the Watch Dashboard, these result sets will be applied every time there is a data update. If there's new publications that match these search criteria, then the user will receive a notification or even an email if that would be requested. Another example of the interactivity and also the way to interactively search and exchange knowledge with other researchers is to add your result set to a collection. And for example, a collection could be shared across specific researchers, or you could also add it to your private collection that you can use for projects and maybe add other information to as well. Of course, at any point, the platform is made that it's all shareable. So at any point, you can share your results, copy this dashboard, share it with another researcher that has access to the platform, and they will receive the same results that you found here and can look deeper into the details as well. Of course, at any stage, you can also dive deeper into the results, find out why this was matched with my complex search query. And also with, of course, linked data, we can continue our search, find progress on the data model, or we can also at any stage dive deeper into the actual source data, for example, going straight to the original publication that's posted on PubMed for NCBI. Good. Of course, we can also, in some case, say, for example, the researcher regarding this topic would like to find key opinion leaders. At any stage, we can find or follow links to the first authors. Could be an interesting start point to find key opinion leaders and receive the contact information for these people as well. Of course, you can also follow to another, many other public information that ONTOFORCE already offers out of the box, such as, for example, all the projects that are related to these subjects, subset of publications that came out of these projects for clinical studies that are conducted. Good, and that concludes my second use case. highlighting a couple of features that are possible within the JPIAMR-VRI Digital Platform. In my last use case, which I will cut a bit short due to the time, I want to quickly highlight the interactivity and flexibility of the platform as well, applying this more towards the data coming from the JPIAMR supported projects and networks. We can do this by going straight to all the data that is already integrated in the platform and receive an overview on what exactly is happening in these projects. The goal here is to, let me quickly refresh. The goal here is to find all kinds of information coming from this data. See that there's a bit of a problem. Let me give a quick refresh. So in general, we want to access JPIAMR projects. And you'll get an overview of all the projects across all of the countries. We can look at the number of projects which are contributed by partner countries. We can also have a look at the sum awarded by countries, for example, and analyze the number of researchers which contributed to this full, to all of these projects. Can create even more analytics, can have a look at the funding by priority area, how it's distributed. And for example, we could also have a look at the cross-collaboration between different partners. Across all of these projects, partners will be working together based on specific topics. Might be interesting to find specific partners, match with them based on your search criteria, and see how they are working together across these projects, and if it might be interested to involve them as well in a new project. At any stage, it's also possible to include new widgets. For example, if I'm interested to see the interaction between partner countries, I can add a quick analytic as well, and I can see how countries are working together across all of these projects. At any stage, I can filter down. It's interactive, and I can find more information on specific topics. Good. So that concludes my last use case. I also want to highlight, of course, that across the platform, there's a lot of supporting material showcased. For example, if we go to the platform, there's an extensive user guide, which at any stage of your step can offer you help, will help you in starting your search, creating queries, finding and manipulating the platform, as well as any other instructions that you deem useful. And then, of course, as mentioned earlier, we have a dedicated help page as well, where there's more information on data model, video help, and much more. We're also open to any feedback at any stage, so feel free to provide feedback at any point. Good. This concludes my demo of some exemplary use cases. We'd like to highlight, of course, that we'd like to highlight that the platform is also accessible, or will be set accessible after the webinar. And with this, I would like to hand over back to Till to continue the panel discussion.
Thank you very much, Martin. Thank you very much. This was excellent again, and I'm pretty sure there is the thinking goes now on in our audience. And if people have questions, we've mentioned it before, but and I see already some questions in the chat. Please put your questions in there. We will have a Q&A session dedicated after the panel discussion, which covers the technical questions, but also the panel respective to the panel discussion. So thank you again, Martin. And this brings me to our next point and in the agenda. And if you could have the slide, please. Excellent. Thank you very much. So I would like to introduce our eminent panel. starting on the left with Andrew MacArthur. Andrew is a chair in computational biology at McMaster University in Canada, and he will certainly speak a lot about the CARD database, the Comprehensive Antibiotic Resistance Database. We have then Charu Kaushik. She's an immunologist by training and the scientific director at the Canadian Institute of Health Research at the Institute of Infection and Immunity. We then have Jean-Yves Madec. Jean-Yves is a molecular microbiologist, and he's the research director at the French Agency for Food and Environmental and Health Safety in Lyon, and he's the chair of the scientific advisory board of JPIAMR. And he's joined by Lucie Collineau, who also will speak about Resopath. Both of them are involved with Resopath. Jean-Yves is leading that. Lucie is a veterinarian from Toulouse Veterinary School and has a PhD in epidemiology. And she is the veterinary epidemiologist in ANSYS. Then we are joined by Rolf Müller. Rolf is studied pharmacy in Bonn and has also a PhD there, and he's a managing director at the Helmholtz Institute of Pharmaceutical Research in Saarland. In Germany. And then last but by no means least is Shoji Miyagawa. Shoji is the managing director at the Department of Innovation and Clinical Research Center at the Japan Agency for Medical Research and Development. And Shoji has many international roles, including the WHO and the UN, and we would be very interested to hear his views, and also from a perspective of the Ministry of Health of the government of Japan. And with that, I would like to bring in our panel members into the call. If you could switch on your videos, then people can see you speaking. Thank you very much. Excellent. And so maybe if we start with the two databases, which have been mentioned already, and if I start with Andrew and get your view, Andrew, on the perspective of the CARD data set and the inclusion in the digital platform, please.
Perfect. Thanks, Till. So hi, everyone. So this is a good day from CARD's perspective. From the day we started CARD, our design principle was to end up involved something like this. We really had a data sharing and a data integration as our long-term goal. So congratulations on the launch of this platform. It's a great day. So CARD as a database really focuses on the molecular determinants of AMR. So it comes from a genomic perspective. So it's a large knowledge base about the genes and the mutations that underlie AMR and how those genes and mutations affect individual drugs. That's really the goal of it. It tracks information around the mechanism, information around the epidemiology, in other words, the distribution of those genes and mutations amongst many pathogens. But really the core is an expert curated amount of information from the scientific literature that's based on peer review on what genes and mutations underlie resistance to individual antibiotics. And so that knowledge base really forms the basis of it. On top of that, we build software for groups, therefore they can analyze their own genomes or metagenomes to make their own predictions. We spend a lot of time working on confidence, how you can be sure that you saw that gene in your data. And it makes a lovely website and lovely tools. But underneath of that, we really talked about what Martin already introduced, which is ontologies, data harmonization and data integration tools. So we spend a lot of time in the background making sure that data and information is shareable between computers as much as it is shared between people. So we put a lot of effort towards harmonization, and that helped power this joint database that's being launched today. But being genomically based, what it really lacks is phenotypic information. Its domain expertise is the genetic determinants, not the phenotypic response of an individual infection. And so I would say it has weak points when it comes to know the MIC for an individual mutation in an individual infection at an individual site in a body. And so that's why integration like this is so important.
Thank you, Andrew. That was a perfect ending, because this shows us the complementarity and the added value by being in the integrated digital platform. Excellent. Maybe I go to Lucie and give a similar question. So where do you see a bit of a background on Resopath and your benefit of being integrated here, or offering through the digital platform, Lucie?
Hello, everyone, and thanks for the opportunity to say a few words on the RESAPAT database. So basically, this data are collected as part of RESAPAT network, which is a French network for AMR surveillance in bacterial pathogen of animals. It's a long-term established network. It has been growing over the years. We now have a network of 70 laboratories that all participate on a voluntary basis. And so the way it works is that whenever a veterinarian asks for antimicrobial susceptibility testing, these data obviously are being generated by local laboratories. And once to twice a year, this data are being centralized at ANSYS. So we collect phenotypic data, primarily SRI data. So this is quite complementary to what Kurt is doing. This is primarily enterobacteria, but we also have other hazards of animal health interest. And we also have metadata, including animal species, both livestock and companion animals, also age group or pathology involved, like respiratory or digestive, for example. So that represents more or less 50,000 strains each year. And for which on demand, we can also have access to the strain that can be characterized through molecular approaches or sequencing for further research or surveillance. So, well, over the years, this data has been quite helpful for our vets to support their decision-making for antimicrobial therapy, but also for our policymakers to sort of assess the impact of our national action plan, because these data are collected very close to the way, like to the point of prescription. And RESAPAT has been pushing quite a lot for data integration. We try not to work in silo just on our own vet side. So wherever it's possible, we try to connect our AMR data, not only with EMU data, but also with AMR data in humans, whenever it's relevant for zoonotic pathogens in particular. And we also try to connect our RESAPAT data with data from other countries. So recently, we tried to launch an initiative called ESVET to develop an European antimicrobial resistance surveillance network in veterinary medicine, where we try to connect our RESAPAT network with our counterparts in Europe that are doing surveillance in animal pathogens. So, well, we really see this VRI platform as an opportunity for us, to connect to other networks. We have done some work already to make our data more visible and accessible to others. There's been a shiny app developed for the RESAPAT data, but it's only RESAPAT data at this stage. And until this VRI initiative, I was thinking that our RESAPAT data was not interoperable at this stage because we have our own ontology, it's all in French and so on. So I was very impressed by all the work that ONTOFORCE has been able to do to sort of connect this data with others, and they've made an incredible job in making sure our data were actually interoperable, and it's much more powerful, I think, what we can do at this stage with the data we are providing. So thanks again for this. I think it's a great exercise, and we are very happy that the RESAPAT data was able to be integrated into this tool. Thanks.
Excellent. Thank you, Lucy. Also, what I wanted to say to our panel, if you want to ask a question in between in the panel discussion, please raise your hand and I will come back to you. Otherwise, I proceed around the table, the virtual table. So we heard about already the data integration and coming from the genomics over to the susceptibility data Lucy just mentioned. The obvious next step would be looking more at the molecules, and I would like to come to Rolf as a drug discovery researcher about your view and ask you about your view on the data integration and the opportunities through the VRI. Rolf.
Yeah, thanks, Till, for the intro. Hi to everyone. I'm also glad this has been opened up now. Let me introduce the network I'm the speaker of for the VRI, I think, which makes a bit of sense here. It's a research alliance for antibiotic discovery and development, and within this alliance we have mostly basic but also industrial researchers from biotechnology to medchem to medicinal microbiology who are en route to discover and develop antibiotics. And that is a scene where you start out from really basic data to target product profiles of compounds. And on that route, you need a lot of data, obviously. So we will probably be more users of the data, and we will be happy that they are available now. There is a lot of data still hidden in pharmaceutical companies, which we are fighting to be able to make use of in the future. But I think it's pretty clear, and I think the presentation that Martin gave presented this very well, that in this process from early discovery to development, we very early need to build in knowledge about resistance and try to avoid resistance or build into the molecules themselves factors that overcome resistance from the start. So this sort of shows how this is all interconnected. And maybe another point I wanted to bring up, the most of our currently used antibiotics are still natural products. 80% of the antibiotics on shelf are natural products or natural products derived. So it's not in the majority the small molecule medchem libraries, which are typically used. And with this respect, as they are natural products, they back connect to microbial resources, which basically make them. And for this, again, the resources earlier mentioned make a lot of sense to be easily available, as we also are very interested to see how the natural products, how they generate not only self-resistance, but also resistance in the environment before the new compounds actually get to be used in human therapy, ideally. Of course, this is a long route. And I think the community that I'm representing here will be very happy with the developments of this platform.
Excellent, thanks, Rolf. And maybe I mentioned at this stage, we start completing the picture, and what is obviously not yet has not yet been mentioned, maybe it has been touched by Lucy, is this is the diagnostics aspect. And because Rolf mentioned the network he was representing, I have been coordinating the network on AMR-DX Global, a network funded through the JPIAMR-VRI, looking at diagnostics and the barrier to development and implementation of rapid diagnostics to tackle AMR, and also about teaching and training aspects. And what we noticed was obviously that there is a lack of available data information about the diagnostics themselves, the rapid diagnostic technologies, their performance and the availability, but also, of course, about teaching and training facilities, which are essential if you want to introduce these. So here, the VRI obviously has a huge opportunity, which we see from that network and from the community we have there. Thinking more global, because the VRI is a global platform, and I would like to ask then Shoji about your experience and your view on the applicability, on the utility of the VRI platform. Shoji.
Yes, thank you, Theo, for kind introductions, and thank you, the JPIAMR Secretariat, for the invitation to the event. I'm very glad to join the launching event. I'd like to congratulate the ladies and the JPIAMR Secretariat for launching the VRI platform. Having seen the presentation on the VRI, I realized that there are great possibilities or the potential in the AMR research activities. I should examine it more carefully, and maybe I could come some more ideas. But so for the researchers in the research community in my countries, Japan, in Asia, I expected the platform would provide a basis to connect our researchers globally and regionally. And it will also be a basis to connect the different sectors like agriculture, the veterinary medicine, medical practice, the pharmaceutical companies, and the private and the public in Japan to global communities. I understand that there are several existing database or data set in the different regions and different sectors, as the previous speaker mentioned. In my country, Japan nosocomial infections database, we call it JANICE, was established almost 30 years ago, and currently almost all major hospitals in Japan join the network and share the data from their laboratories. And there is a data set from veterinary sectors in Japan too. I hope that VRI could create a research hub to get together those currently existing AMR network and data set or the database in various regions. So I'd like to stress this is quite important to share the information, the data, and I hope the JPIAMR and its members promote this platform to utilize the platform as much as we can. Thank you.
Thank you very much, Shoji. And you mentioned, obviously, but this is very important aspects you brought up, and brought up a very important aspect which I want to put to Martin in the Q&A. And I saw also there was a question about interoperability and how this is ensured and accomplished. That is something which I certainly would like to have a discussion on in the Q&A for sure. But also about there are so many data sets which are currently not in the VRI. How is the process of entering them? How would people who may represent data sets and databases approach the VRI and bringing them to into the platform and make them available and integrate them with the rest of it, so for the added benefit as described. The other aspect which you mentioned, so we come back to that in the Q&A probably, but the other aspect you mentioned was about the data sharing, the global community and the benefit for the global community. And we are all having this meeting now virtually because of the global situation with COVID-19. And we learned a lot during COVID-19 about the benefit of data sharing. And I think it would be good to reflect a moment in the panel on the learnings we had from the COVID-19 pandemic and how we could translate this to the silent pandemic of AMR. And maybe I come to Charu first, and if anyone else wants to come in on this, very happy to pass on to you. So Charu, what's your view on this, please?
So thanks, Till, and I'll start out again by congratulating the whole team, the whole JPIAMR team and the researchers and those who've contributed their networks and their databases. It's a good day for AMR community. I bought into this dream 3 years ago when I started this job, and Edith had already been working for 1.5 years on it. And I bought into the vision because I realized that, being a researcher, and I'm sure, most people here are researchers, that research is only as good as its use. So having research sit in publications and sit in our laboratories. So this is a perfect example of knowledge mobilization, getting your research and getting information to those who can actually use it. So I think from my agency's perspective and JPIAMR, this is a huge part of the accomplishment today, is to actually get knowledge into the hands of those who want to use it. So congratulations to the team. And then in terms of what you're asking, Till, about lessons learned. So the first lesson is that, before this pandemic, I think this pandemic has been, shown remarkable progress in our ability and our willingness to share data. Right from the beginning, where the sequence of the virus was put online and used for rapid diagnostics within 2 months of even figuring out what this virus is, to being able to diagnose it, to be able to have rapid diagnostics that were used worldwide now. Here we are, 15 months. It's been a long time. It feels like a lifetime ago that we were seeing each other in person, but it's remarkable that we have 6 excellent vaccines. And that accomplishment would not have been possible without the willingness of people to share data. So the achievements, of a global community, because we really are a global community, even though the national boundaries exist right now, is really about how easily the scientists and researchers and the global community was able to share the data. So that, I hope, is an irreversible process where people's willingness is going to continue. You know, many of us were talking about AMR as a slow-moving pandemic. You know, so hopefully this pandemic and the implications of what has happened, and we've seen it change our lives, will put some impetus into having that willingness for AMR data as well. You know, you can hear from the excitement in those whose data is being shared in this phase 0, how much it means to this community to be able to share that information with others and have it be used by others. So that's the silver lining. You know, the pandemic has also then highlighted some of the barriers that continue to exist. You know, so I will cite a few issues around, you know, the FAIR principles, the interoperability of the data we just talked about. So we are seeing this in spades in national level, where even in Canada, you know, the interoperability of data between provinces in the context of COVID, you know, is a huge barrier. And then newer things that we are hearing in terms of IP issues. Of being able to share data and know that, you know, you can still own the data. Huge issues for LMICs in terms of data ownership. You know, who owns the data and how it's used. So there are definitely barriers, but the hope is that through this painful experience of last, you know, 16, 17 months now, you know, we've learned how we can do better. And there's hopefully more willingness on behalf of funders, governments, researchers to then move forward to learn those lessons and apply them to AMR, because again, you know, data has to be made visible and shared for it to be of any use. So hopefully that addresses some of the questions that you were asking.
It does. Thank you very much, Charu, that excellent answer and very comprehensive. And I'd like to pick up one point you said about of course, the lessons which we learned through through COVID and the slow-moving pandemic with AMR. Maybe AMR was just, or for sure it is, one of the issues is it's slow-moving and it's too slow to see. And with COVID being so rapid, people immediately saw the benefit of sharing and the benefit of interventions. And that is something which we are now building on. I hope we will be building on, and we see this already, and including today, that sharing of information, sharing of data will advance the field. I don't see any hands, so I would move from here in the thought about, thank you, Charu, again, and towards the other aspect, which also was amplified in people's perception through COVID, was One Health. And we all know already that AMR is a One Health issue. And as I said, with COVID, this was amplified again. And I would like to ask Jean-Yves about his view on from a One Health perspective and the data sharing and the VRI, what it helps addressing the issue of AMR in the One Health perspective. Jean-Yves.
Yes, hi everyone. Thanks, Tia, and thanks everyone for this this presentation. Yes, it is a great day, of course. And first of all, I would like to thank all of those who have contributed to this platform, and of course ONTOFORCE. It's an amazing job, and also of course JPIAMR, and in the person of Edith, who was very much engaged. Yeah, you're right, Till. It's from the first day. So yes, it's a great day, and of course I will not emphasize too much on the importance of the One Health perspective, because we all are convinced of that, I think. But when I can just come back on this slow versus rapid moving of themes or research topics. We can have also in mind the cholesterol issue. It was not that slow. It was a very important at international level crisis on the AMR topic, I would say. And most probably, most likely, we would have got a lot of benefit of having such a digital platform to share information. And most probably the issue would have been at least not solved, but documented not in months, but probably in weeks or days. So sometimes AMR is not slowly moving. Another example was this MRSA crisis in the Netherlands, early 2000s, when a very new MRSA type has emerged in the pig sector, causing disease in humans and pig farmers and all professionals related to pig production. So most likely we will have other situations like that. And I think the One Health importance of how we look at this topic, the AMR topic, can learn lessons from the COVID as well, even though it's not a pandemic, strictly speaking, but or a slow-moving pandemic. But we have a couple of examples in that sense. Another point that comes to my mind is we were talking about genes and relationships between genes and phenotypes, but we have also major relationships between antimicrobial use and antimicrobial resistance. And we all know that the central question is whether we have an impact on one sector when we act in another one. When we reduce antibiotics usage and prescription in the animal sector, what impact do we have on the human side and vice versa? And I also think that part of the answer will come from extracting data from large shared data sets and then analyzing them together to see whether we can find some clues at least, because this is at the moment still unanswered questions. And so when we have, for instance, in France, we have nice examples of strong decrease in the exposure of animals with antibiotics. We don't see anything in the human sector. So does it mean that this One Health perspective is not that true, or does it mean that we don't have the clue to decipher the tiny roots or the real driving force, which may explain the interconnection for the AMR issue between the different sectors? I also think that we should more connect AMR data sets and non-AMR data sets. For instance, what I can observe usually in the human sector, that people do not necessarily see how it is important to consider the distribution of the different animal species per country, for instance. Not all the countries, when you're looking for the human population, of course, the human population is more or less distributed with the same driving factors or parameters in each country, but this is not at all the case for animal species. And most probably AMR does not transmit equally when we're talking about cattle, pigs, companion animals, aquaculture, or other kind of sectors. So in a One Health perspective, connecting non-AMR data set and public data, as it has been done in this platform, is highly important, in my view. And yeah, finally, also when we're connected data to data and data to people, it's also important to connect people to people. And in a One Health perspective, if we want people to work together and share data, we must connect them together. So we must connect just simply the research communities, and the huge barriers or really room for improvement to connect veterinarians with human doctors, and also people from the environmental sector, which is a huge sector. And when you talk to those people, well, there's a completely combination of different people, different skills, experience. So it's highly important for this as well to work into One Health perspective, to connect all these different parameters and data sets and people and sectors. A lot to say.
I I'm sure we could continue. Thank you, Jean-Yves. We are coming to the timing of the Q&A. Before I wanted to have one more question, at least here, but just to remind the audience that you can put some questions in the chat and we direct them to the panel. What I wanted to pick up from what you said, Jean-Yves, about the intersectoral integration and the obvious need to do that, and the different natures of data. One aspect which we have spoken about, interoperability. What I always think about is if I look at databases, this is the effort which it takes to keep them alive and to keep the data integrity and alive, and have sustainability of the data information. And probably Andrew is a good place to ask him. How about your experience with this, and where do you see also the challenges for the digital platform from this respect of keeping the data up to date?
Yeah, I think there's a lot to say. I think, and we have to highlight how difficult AMR in particular. Like, we sequence the human genome, that's a static data point that we just layer new knowledge on top of. AMR is a moving target. So a database that is accurate mine yesterday is out of date today because there's new mutations and new genes, especially when we pay attention to veterinary environmental sources. I'd compare it to parasitology in the 2000s. The parasitology, I worked in that field. We sequenced trypanosomes, Plasmodium, Giardia, and there was this rush of money and data. And in a short window, there were many databases and many software platforms, and very confusing for the community, very much what AMR is right now. There's dozens of databases like mine and tools. But parasitology consolidated and matured. So now when you look at that community, there are standards, there are operating principles, there are less data sets and more integration. That's really what we have to drive for, which means what you're going to see is less boutique databases. And by definition, that means you have to focus on interoperability, that of value beyond just what you do, that it can be integrated into efforts. And this effort and others is amongst the first to really mature the community. What that means is a large focus on standards, both, broadly, say in bioinformatics in my case, but within the community of standard practices, but really a high focus on trust. Really, if people are going to use it, your data points, your predictions, your measurements have to be all of very high trust. The practical aspect is every government has different approaches. In America, you often get your database funded when you're at the bench, and as soon as you put the pipeheader down, the database money evaporates. Other places have funding mechanisms for long support of infrastructure, and they think in a 10, 20-year window. So you do have to have a business plan as a database. And I think as a result, you need to focus on your domain expertise, what you do best, and therefore let others do what they do best and do integration like these. There has to be a push in the community that funding for initiatives like this, funding for individual efforts that compose this, that they get steady funding. And that a long-term, this, as we say, this is a long-term battle. It is a slow pandemic in some ways. So 5-year funding for a database effort is insufficient. There has to be stability to it. In CARD's case, we do some commercializations to sort of stabilize the budget, keep that going. And then as we become more and more global, there's just the sheer demand of infrastructure. So I'm in genomics, which is a very expensive field. So keeping millions of dollars of computing to keep the database fresh is a major undertaking. These are big challenges. For it to work well, you need to have value. And so if CARD was just its own little boutique effort that pleased public health officials, I would be okay. But the fact that CARD can integrate by using standardized processes, data sharing algorithms, listening to the computer scientists as much as the public health officials, that allows it to be high value and to contribute in ways that I never anticipated, like this one. That's really where we have to go. That means as researchers, as grant panels, as paper reviewers, we have to really push on that standardization and interoperability. Funding agencies will respond to that you're making that effort, and that will provide stability to us. But overall, biocuration, the construction of databases, as I said, is very labor intensive. The field is fairly young. There's a good society driving into it, but a lot of funding agencies have a lot to catch up to make sure we have this stability.
Thank you, Andrew. Excellent. Very, very important points. And I would like to hand over this question also to Jean-Yves or Lucy about ResaPath. How you see this in terms of sustainability and your business model. Jean-Yves, yep.
Yeah, well, you're right. It's a highly important point. The ResaPath is based on a win-win model. I mean, it is, as Lucy was mentioning in her presentation, it is a voluntary network. So people are including in the network if they want to and if they get the benefit from it. So it's highly important that each party gets a benefit from the other one. So the deal, which up to now has been highly successful in the sustainability of the network, was that ANSYS as national agency has the opportunity to get data, to gather data from AMR data from field conditions and to provide trends which are highly useful to the French Ministry of Agriculture. So this is the benefit from ANSYS, from globally for the administration. And the benefit back for the local partners is that ANSYS has put a lot of efforts to help them to provide a good and accurate data to their clients, because this is based on the private system, because this the connection between veterinarians and veterinary laboratories are from the private domain. So the objective is to make those laboratories keen to provide the data, because they get a benefit back from us. And what we are ensuring is that when you are part of the network, you have more chance to do a better job and to have more clients and to give more reliable results and data to your clients. So this is a win-win model. We call it a win-win model. And for now, it has been highly sustainable. Sorry. I don't know, Lucy, if you want to add something.
No, I think it's a great summary. Just to say that we're going to expand in the coming years. There are, I think, more or less 30 labs still waiting to join the network. And we're limited by data management capacity, which have improved recently. So, we just expect our network to continue to grow in the coming years, which is quite encouraging.
Yes, we have the other problem, having too many client members.
I think one of the things we learned is that you do have to be cutthroat. You need to pay attention to what's useful to people, investigators, clinicians, public health agencies. And if you started an initiative that's taking staff time and it's not useful, no matter how proud of you are of it, you got to cut it out. And that's the hard lesson we've learned about that. If it's not high trust or high use, then it's merely academic, and that's not enough.
Maybe that's a good point to also ask from a global perspective, also Shoji, from your perspective in terms of sustainability and funding such initiatives, where do you see the opportunities there, or the challenges as well?
Well, this is a very difficult question for everyone.
Exactly.
So yeah. So of course we have to perform or show that our value to the stakeholders, and then they try to get the resources from government, and but so the maintaining the database or the data set is a very, very difficult issues. So maybe nobody can answer to the correctly that maybe maybe I could say that the JPIAMR is a broad network of the funding agency and the research institutions. So therefore, that's maybe we could cooperate each other. So sometimes some funding agency supported some part, and some other funding agency supported. So maybe we could cooperate to help the maintain or the develop our database. Thank you.
Thank you, Shoji. And I think we as a collective group, we all see the value of this, and the value will be demonstrated also and the impact as the platform proceeds. But obviously, as we have now said a number of times, all of you, that this is a challenge to keep things afloat and keep them updated. Anyone comments on this from the panel? Please raise your hand. If not, I would like to, there are no questions in the chat in the moment, but I would like to bring in Martin because there were questions in that direction in terms of the technicalities of adding more data sets. So what would people do if they had one of these data sets Shoji was describing, for example, in Japan, if they wanted to join?
Yeah, thank you for that question. I will quickly try to summarize that. So in general, the platform upon which the JPIAMR-VRI Digital Platform is built allows for very broad integration possibilities with other datasets. So if there's a dataset available, this can, of course, be integrated and we can connect this up with the other data sources as well to make it as shareable and interoperable as possible. So I hope that answers the question.
If I don't see that answered the question to me, but if there is any further question to that, I would like to ask the people to put it in the chat, but I don't see something, but I saw Edith came up with her camera, and I'm sure you want to also respond to that.
Yeah, I just want to add that, and it brings back your question, Till, earlier, that in the future, with the next step going forward with the VRI digital platform, obviously we are welcoming all suggested new data sets or data sets that we could include in the next phase. So we will look into it and make assessments of various criteria for those new data sets, and obviously we'll consider them. So please do comment with the comments and suggestions through the platforms or through the JPIAMR Secretariat directly, and we will be happy to look into it for the future.
So Till, can I add to the
Go ahead, please.
Discussion? Yeah, so just in terms of the question that you asked, Shoji. So Shoji and I wear our funders hat. For funders, there's only two ways that we can support long term. Either it's clear that it is an infrastructure that is critical for moving the field and keeping it sustainable. Otherwise, competing priorities take over. If funders could fund everything, every time we started a network, we could fund it for 25 years, we would never fund something new. So, so researchers and we ourselves struggle with that to say, this is great, this is priority, we need to do this, but how long can you maintain it? So we always start networks by telling people you have to build a sustainability plan. And that's, where Andrew and Jean-Yves and Lucy are talking about that if there's a value added to the community, there needs to be some kind of sustainability that can maintain it. So there's that proposition. The other is that if there's commercialization, which unfortunately in many places, when you fund things that are at a basic science clinical level, we don't think of the other end of the pipeline, neither do researchers. So I think that's really, really important to start integrating that piece into when you develop something. And I think Andrew alluded to it, that you have to make some tough decisions where the transition has to happen from academic to what will it mean to keep the funding coming, not from government sources, but from commercial sources, which make use of the databases. So I think those are two principles that would be really, really useful for everyone to remember. And of course, this has been a labor of love because there has been no money for this. So the phase 0 has really been a labor of love on behalf of many of the funders, just to see that what use can the community make from this if we were to build it. So it's built. I hope the community puts it to good use so that there is reason for funders to say, we need to keep this sustainable.
Excellent. Thank you very much, Charu. Andrew, we are up. Time is up, but your hand is up. So very quick response.
Sure. We do a lot of commercialization to fund the card. And I think the fundamental point I've learned from that is, AMR is multifactorial. We need academics, public health, government, but we really need industry of, food sector, pharma sector, and all the rest. And licensing is very important. Licensing to a company is a promise of quality and return of effort, as opposed to a website where you just download for free. So I thought industry would be reluctant, but they really like that framework. Yes, we make money off it, and that pays for my developers and keeps card running. But it also, that license is a promise, and that engages industry in the fight against AMR. So I think it's a really important thing to consider with our database efforts.
Excellent. Thank you, Andrew. And this is a very positive outlook. And I would take the opportunity to stop at this point and stop by thanking all our panelists. So thank you very much, Shoji, Rolf, Lucy, Jean, Yves, and Chao, and Andrew. And hand back over to Laura to conclude the session. Thank you very much.
Thank you, Till, and thank you all for this fantastic panel, this presentation. It has been really, really interesting discussions and also a lot of food of thought for the future and what can we do for next stages. So thank you all for today, and also thank you all to has contributed. There's so many people who has contributed to make the VRI digital platform today happen, especially to the team and ONTOFORCE, to Siobhan, to Edith, to all of you. And just to finalize, just to tell to all the participants, thank you for being here today. And please go on, log in the platform, start using it, share it with your colleagues, share it across, because we just want it to be useful of value to the community. And please send us any feedback as a user that you might have. So please log in, enjoy the platform. So thank you all, and goodbye.